So I'm going to talk about how neuronal networks, so brain circuits, solve problems that an animal encounters in its world. And much of what we do, like Sitabra mentioned, relates to two what we think of as paradigmatic systems in neuroscience. One is the olfactory system, both the insect and the mammalian olfactory system, and the other looks at navigation and to some extent memory when we think about the hippocampal formation and the entorhinal cortex. So today what I'm going to talk about is looking at the olfactory system, and it will give you a sort of broad sense of some of the kinds of models that we make when we think about these systems, going from very idealized point neuron, integrate and fire kind of neuron models, all the way to biophysically detailed networks of neurons. So I actually made this picture using ChatGPT. It's quite remarkable that you give it a prompt and this is what it comes up with, right? Sort of encompassing a couple of things that we'll talk about. One is this plume, not exactly a turbulent plume out here, I would say, but nevertheless a highly variable plume that an animal uses to navigate through its world, right? So we'll touch on multiple aspects of this. Now, I feel free to stop me anytime during the talk. We don't really have to get to the end of the talk. We can just potter around somewhere in between too. And
Just curious, what prompted you to
Oh,
get that
I
picture?
actually, the prompt I gave out here is of a wolf navigating a lush green forest following a following an odor plume, right? And so there's an interesting story why I chose a wolf, because one, I have a dog, so it makes me very close to wolves. But the other thing we discovered that our dog is sort of part dog and part wolf, because Pune has this Saswad grassland. So this was an indie that we got off a coffee shop. And when Uma's group in NCBS, they discovered the first cross between wolves and dogs in Saswad in Pune, and we'd been suspecting, oh, this one is not really listening to any commands or anything of that sort. So we sent a picture of her to Uma, who checked with her canid expert and said, that's almost 50% wolf. So we've been keeping children away from her because she thinks children are edible. No, I'm just kidding. But nevertheless, so therefore the wolf in the prompt. So there are a lot of stories within stories, wheels within wheels. But anyway, in case I don't get to the end of my talk, I'd like to acknowledge the people that I work with. So much of what I talk about today is done with three students, undergrads in my lab, Rishika, Sandeep, and Pratyush, who worked on different aspects of this problem. And there are a couple of graduate system students, Siddharth and Gaurav, who now work on other aspects of the problem. So a lot of what I'll talk about is really work that these guys did over several semesters in the lab. All right, and some of my collaborators and co-conspirators and funders who've been generous enough to give us money to do the things we enjoy doing, to no financial gain apparently, but nevertheless. All right, so when we think about so if you look at all sensation, you look at perception, you look at action, a lot of it is driven by the activity of networks of neurons, right? And what we really care about when we look at these networks of neurons is the spatiotemporal patterns they generate, right? So when you smell a rose, there is a group of neurons that fire. There are specific neurons will fire at specific times, right? So there's a spatiotemporal pattern that represents in your brain the smell of a rose, right? It's not that one to one, but nevertheless, for for practical purposes, let's stick with that. And what I found looking at all of this spatiotemporal patterning is really one of the first systems that one really needs to think about when we think about how these patterns emerge is really the motor system, right? Because turns out that and I'll give you an example of that. Turns out that if you take movement out, you don't have to move. We wouldn't evolve brains if we didn't have to move. But let's, rather than looking at an organic system, let's look at an inorganic system that generates really remarkable patterns of activity. And this is an automaton which you will find in so if this movie actually did run, it's on YouTube. So this particular automaton is what's known as, it's known as Maillardet's automaton. It's a 19th century automaton that you will see in the all right. So anyway, I'll describe it in words. So this particular automaton has, it's got the visage of a child. It's got articulated hands. And this is in the Franklin Museum in Philadelphia. And so somebody dropped this automaton off out there somewhere in the early 1900s. And yeah, there. So this particular automaton, also called the draftsman writer. So if you stick a pen in its hand and put a paper down, it's going to make these elaborate patterns, right? So it makes a bunch of these images, maybe 4 or 5 of these images. It writes down 3 poems in this beautiful calligraphic hand. And you'll also see that they were very careful with how they had the head movement and all programmed in, right? Now there's no electronics in here, right? This is just a mechanical device. And at the heart of this automaton, you'll find these circular discs, right? And there are indentations on top of the discs, and there are these arms that sort of go up and down based on the indentations of this disc, and that's what gives rise to these beautiful patterns. Now what's really interesting, of course, is that so these patterns are generated over a time scale of approximately a minute or so. It will do this entire drawing, and you'll see no obvious periodicity in there. Nevertheless, if you look at the insides of the automaton, you have hundreds of these discs, right? Basically oscillators, and you can use these periodic patterns to generate aperiodic, of course, over a certain time scale motion, right? And I sort of thought of this as really interesting, and of course it's not surprising because Fourier had the same sort of idea when you can generate arbitrary patterns in 1 and 2 dimensions. This is something I took. These are called Fourier epicycles. So you can compose a bunch of oscillations and you can generate arbitrary patterns, right? So if you look at the brain, now it's composed of millions and millions of neurons, and neurons can behave as oscillators. They can behave as a lot of other things, right? But, and we'll talk about the other things that neurons can do, but as a first approximation, for starters, let's think of neurons as oscillators. And one of the first questions that one can think of, inspired by these motor systems, by these motor networks, is how do you get this bank of oscillators to generate arbitrary spatiotemporal patterns? Patterns that could represent an odor, that could represent a thought, that could represent a ballistic movement through space, perhaps bird song. You can have all of these patterns, and how can one compose oscillators that you see in the brain to come up with these spatiotemporal patterns, right? So that's sort of the agenda that we'll look at. So if you look at neurons in the brain, so here's an example of a layer 5 pyramidal neuron, and depending on the input you give it, it can fire at different frequencies, right? So you have an oscillator of this kind. Not only can single neurons behave like oscillators, you can also connect them and still generate oscillators. One of the classic oscillators that you see in movement is what's known as a half center oscillator. You will see some of these in your spine. You will see these oscillators driving periodic movement. So if I had to move my finger periodically, I'll have to compress this muscle, which is a flexor, and then alternately compress the other muscle, which is an extensor. So you need neurons to fire in an alternating manner, and that's often driven by neurons that are coupled like this. It's called a half center oscillator, right? So these neurons are essentially inhibitory neurons, right? So a caricature of an inhibitory neuron is that when one neuron fires, it's going to prevent the other neuron from firing, and vice versa. So that sort of behavior is what gives rise to this alternating kind of a pattern, right? If you look at a rhythmic inhibitory circuit, in this case in the lobster's gut, right? So here you have a network consisting of 3 groups of neurons, and you'll see that they fire in alternation, and the fact that they fire in alternation is strongly dependent on how they are connected to each other, right? So that determines the phase of these oscillators. So single neurons, networks of neurons of different sizes, all of this can generate oscillators of oscillations of different kinds, can behave as oscillators, right? Now the output of these oscillators goes to the effectors, right? So that goes to your muscles. In the case of jellyfish, you can see that this is actually not a half center oscillator, but it's rather a nerve net, and you have this alternating pattern of, not alternating, you have this rhythmic pattern of activity that generates this sort of a movement. You have a bunch of half center oscillators that give rise to movements like that in the horse, and horses have different gaits, right? So the reason I put this image out here is that if you look at the phase relationship between the legs, right, what's different between these the trot, the canter, and the gallop, different sort of movements of the horse, is the phase relationship and the timing of these oscillators. It's still the same muscles that are being activated, but the phase in which they're being activated is what changes as you go from one to the other. So the same network, and we know this also from other networks, the same network can have multiple phase relationships between these oscillators, and each of these phase relationships can be then translated to some behavior, can be mapped to some particular behavior, right? So a lot of what we do, I think, is motivated by movement, and it is really an attempt to think of the cortex, various networks in the brain, as central pattern generators. And Daniel Dennett couldn't have said it better. So here's an example. So you have a juvenile sea squirt. It wanders through the sea searching for a substrate to cling on to and make its home for life. Once it does that, so it has a ganglion equivalent of a brain that it uses to sort of go around its environment, and once it finds a spot and hangs on to it, it doesn't need a brain anymore, so it eats it. Apparently this is rather like getting tenure. Right? So okay. Nevertheless, so the idea out here is a lot of what we do is essentially what I think of as an ode to the primacy of movement, right? Movement gives rise to these networks that you can that are recapitulated in lots of different parts of the brain. All right. So before I get into thinking about olfaction, okay, let's pass on that. So before I get into thinking about olfaction, I'm going to take a slight bit of a tangent in order to define a very simple ideal, an idealized model network. And once we have that, then we can talk a little bit more about olfaction and see how we can break it up, right? So bear with me for maybe 10 minutes, and then we'll get back to to olfaction. All right. So here's an interesting observation, right? So you have two neurons, all right. They inhibit each other, right? So their interaction is antagonistic. There's another person I know. Their their interaction is antagonistic. So when one neuron fires, it prevents the other from firing, and vice versa. So that's what gives you this half center oscillation. It goes back and forth. All right. Now there's an interesting sort of construct that one can relate it to, and that's what you can think of as graph coloring, right? So what's graph coloring? You can think of it as a prescription where if two nodes of the network are connected to each other, then you have to assign it different colors, right? So that's the prescription. And for example, if I take this particular random network, I know that this node and this node are directly connected to each other. All right. So I have to assign it different colors. So here there's a light blue and that's a red. Okay. Now, if they are not connected to each other, they can be assigned the same color. It's not necessary, but they can be assigned the same color. So graph coloring is basically this prescription, right? Now you can see what the connection between these two are, right? If two neurons are connected by inhibition, they won't fire at the same phase. They'll fire at different phases. If they are oscillators, they'll do qualitatively different things. And if all of them get the same input, then perhaps one can map the coloring of the graph to the activity of the neural network, right? So perhaps all the reds would fire synchronously, all the greens would fire synchronously, and they'd fire in different phases with respect to each other, right? So let's sort of start off by making networks. Yes.
Assumption about the network?
Sorry?
Are you making some assumption about the network, like the right type of interactions?
So here the interactions are all inhibitory, right? So I am making an assumption that the interactions are inhibitory. I'll get to a point where we need excitatory interactions also in order to set up a network that fires in a manner that it respects the coloring of the network. Okay, right? Okay. Okay, so let's start off by constructing a network which has a predefined coloring, right? So this is a three-partite network where you have three groups of neurons that are all-to-all connected across. So that's the ones in the off-diagonal elements, but within each group they're not connected to each other. Right, so it's a three-partite network, and I've just colored it orange, green, and blue. Right now, what I've done is I've stuck a neuron at each of these nodes, and these neurons are connected to each other via inhibition. In this case, these neurons are actually really detailed, biophysically detailed neurons that you find in the olfactory system of insects. So it's curious that it does some of these things. And the connections are inhibitory. They're what are known as fast GABAergic connections that connect these neurons. And this particular plot is what's known as a raster plot. So on the x-axis you have time, and on the y-axis, every time you see this little line out there, that's the time at which a neuron, which is indexed on the y-axis, generates a spike. All right. So you have group of orange, green, and blue neurons. This is of course a symmetric network. So you have I think 10 orange, 10 green, and 10 blue neurons, and I turn it all on, right? Everything's getting a DC input. When I give it a DC input, you'll see that they all fire in this alternating antagonistic manner, and you can get a sequence that's generated, right? So there seems to be some mapping, at least in this simple case, between the coloring of the network and the activity of neurons. Yes?
You said that same color neurons spike together, right?
Yes.
So is there anything that ensures the synchronization?
So that's a really good question, and I will. There are things I haven't told you yet. There is excitation that we use to synchronize things. So I will get to that in a little bit. Yes. All right. So now neurons. So this particular network that we have, it's a k-partite network. It has a single coloring. Yes.
Could you specify those inhibitory connections? Is it from one neuron of a group?
So it's from one neuron to all the other neurons. So that's the ones out here. But there are no connections within the group.
Okay, and his question was, how do they synchronize if
Yes.
they're not connected?
Yes. Right. So it turns out that one way to synchronize is to connect between groups, and typically in arbitrary networks it's very hard to say that this is a coloring that I want, right? So you can have random excitation across, and that will tend to synchronize depending on the balance of excitation and inhibition. But we'll get to that in a bit. All right. All good so far? Okay. All right. So that particular network has a single coloring, right? There are green neurons, there are orange neurons, and there are blue neurons, and you can't sort of move things around, right? But in general, networks need not have a single coloring, right? Graphs need not have a single coloring. There can be many ways in which one can find what's known as a minimal coloring for a graph. What's a minimal coloring? It's basically the minimum number of colors that one needs in order to completely partition the graph into different groups, right? And there can be more than one way of doing it. So for example, if you take this, so some of these are equivalent, but you take this set of neurons, the three, we know that they have to be colored red, green, and blue. So they have to have different colors because they are all to all connected. This guy we know cannot be colored green, but it can be colored red or it can be colored blue, right? So there is this ambiguity, and this ambiguity, it turns out, has dynamical consequences as far as the network is concerned. So what happens is that when you have multiple colorings, you can have many different things happening. We'll talk about it. One of the things is that the network can fall into a solution where only one of those colors is expressed in the dynamics. Then you can perturb the system and it will go and express a different coloring, right? So you can get various patterns. Right. Now, I can sort of construct these k-partite networks. I can construct simple networks that have a particular coloring or multiple colorings, but these are really very simple systems, and what we really wanted to work with are systems that are far more complex. And that's where we started thinking about how do we construct networks that have a lot of different colorings, but we know these colorings a priori. And one really good system turns out to come from the game of Sudoku. Are you guys familiar with Sudoku? All right. So Sudoku is this game where you have this grid. Typically, it's a 9 by 9 grid, and you have 3 by 3 blocks in between. And it's literally, I would think of it as an accounting game, because what you need to do is make sure that the numbers 1 to 9 occur once in each row, each of the columns, and each 3 by 3 subgrid, right? So you start off the game by giving a certain number of clues. It turns out that there are some theorems now that say that you need perhaps a minimum of, I think, 16 in order to have a unique Sudoku solution. It doesn't guarantee it, but it's a lower bound. Now, if you put a certain number of clues out here, the solution to this you get is unique, right? Now, let's start off by taking some of these clues out. Right? So as I take some of these clues out, the number of solutions that are possible will go on increasing, right? Because you're reducing the number of constraints. And when you take out all the clues, it tells you how many possible Sudokus there are, and that's this astronomical number, right? So that's how many possible Sudokus there are, right? Now, which is why people play Sudoku, because there are so many different ways in which you can arrive at a solution. Now, how does that relate to the coloring? Well, each solution of Sudoku turns out to be the coloring of a particular graph, right? So here's what we can do, right? So let's say that each of these are the nodes of a network, all right? And if I connect the nodes such that each row, each column, and each 3 by 3 subgrid are all to all connected, right? So that's the constraint that I put on the graph. Now, if you were to find a coloring of this particular network, then each coloring would be a solution to Sudoku. And the reason is, if you look at all the nodes in this particular row, right, they're all to all connected, so you have to assign it different numbers, right? If they're connected together, they have different colors, numbers, same difference, right? So you have to assign different numbers to every element in each row, each column, and each 3 by 3 subgrid, right? So if you have a graph that's built like that, then every coloring of this graph is going to be a Sudoku solution. Now, how does this map back to our neuronal networks? Well, I'm going to stick neurons in each of these nodes, and the connections between these neurons are going to be inhibitory, right? So they are antagonistic connections. All right. So if you have antagonistic connections, you know that those neurons cannot fire at the same phase. If it's an oscillator, they're going to fire at different phases under certain conditions. I have to tell you, I'm not a mathematician, so I do a lot of hand wavy stuff. I put a lot of things under the carpet, and it's your job to discover them. I'll just move on, right? So nevertheless, so these neurons behave in an antagonistic manner, so they're going to fire at different phases. If you assign each phase to a color, then you'll end up with a coloring which is equivalent to a solution of Sudoku, right? So let's see if that actually happens. And in order to do that, I'm going to, rather than look at really complicated Hodgkin-Huxley kind of conductance-based neurons, I'm going to think of neurons as phase oscillators. These neurons are what are known as integrate and fire neurons. So if you give it an input, it's going to go up. The membrane potential is going to go up. When it reaches a threshold, it will fire a spike and is reset, right? There are also oscillators known as Mirolo-Strogatz oscillators, and that's the equations that I use, but they're equivalent to integrate and fire neurons. So the idea is this, right? So you have a neuron go around in a circle. When it comes here, when it crosses a particular phase, it fires a spike, and of course your phase is set to 0. Now, if you think of two neurons that are connected to each other via excitation, right? So what's happening is this neuron excites this neuron, and this neuron excites this. So it's reciprocal excitation. When you do that, so when this neuron fires a spike, so the top neuron fires a spike, the influence of that spike is really to increase, is to move the phase of the second neuron a little further ahead, right? So it's going to hasten the timing of the spike of the next neuron, right? So this neuron then spikes a little earlier than it would have had it not received this excitatory input, and vice versa, right? So when this spikes, there's some input out here. Same thing, right? When this neuron spikes, there's an increase in the phase out here, and it hastens the onset of the spike in the second neuron. Now, if you have inhibitory neurons, rather than, so when this spikes, I should have had a picture for that, the phase, rather than increasing, my hands are so shaky, but the phase, rather than increasing, would decrease. Yes, I do, but my laser pointer, like my argument, is all over the place. Nevertheless, so so if this were an inhibitory input, then the phase of the neuron would actually go down, right, rather than up, and it would sort of make the next spike occur a little later. So they have different consequences. So here's an example of an integrate and fire neuron. Here, these two neurons, one that generates these gray spikes and the other the black spikes, as they go on, if they're connected by excitation, they'll tend to synchronize. If they are connected by inhibition, their phases are going to separate out, and they are going to go into an anti-phase kind of an oscillation.
Is this discrete in time? Is it a map?
Well, you could convert it to a map, but this is continuous. It's a simple ordinary differential equation with a reset in it.
But the hike is the
So when it reaches this point, we just say that it has spiked, and then the reset is that you bring this phase variable back to 0. Right, so you can map it to a circle, so it's all fine. Yeah. Otherwise, you can just reset it arbitrarily. Okay, so that's what you would get if you had inhibition. The phases would tend to separate out, and so if I had 9 groups of neurons, all right, and I connect them all by inhibition. It's a symmetric group of, so it's a 9-partite network. I connect it all by inhibition. Their phases would all separate out, and you'd get this kind of a splay phase state where all the neurons are firing at different phases of the oscillation, right? So this is when you have 9 neurons all connected by inhibition. Okay. Now, so here's where we start off, right? So this is just a simple simulation with a large number of neurons that are connected via inhibition. Actually, this is a network that's connected using the constraints of Sudoku, and then you eventually end up with these reorganizing into 9 groups, right? And these 9 groups have different phases, and they'll go around in a circle, right? Now, this particular example is one coloring, right? Now there's another shuffling that I can do, and I'll get a different coloring, and I'll show you what I mean by that, right? So this is okay. So here's a Sudoku which had a bunch of which had some clues, and this is the solved Sudoku board, right? So this is the solved Sudoku board, except what I'm not telling you is that this was solved using this oscillator network, right? Or this is the solution that it landed up on. Now, if I, so out here, there are 81 such oscillators that are connected, constrained by Sudoku. And these blue dots that you see, that's the time when the neuron spiked, right? So that's when this integrate and fire neuron generates a spike, and on this axis you have time, right? So I'm going to take a thin sliver of time out here and see which neurons fire at that point in time. Okay, so this is labeled 1 to 81, and the labeling out here is 1, 2, 3 all the way to 9, 10 to 18, and so on all the way to 81, right? So it's MATLAB labeling, so to speak. You go vertically down the matrix in order to increase the index, all right? So the first element out here, so that's labeled 1, that spikes out here. So that is a 3 in this particular network. Now let's look at the next row. You have a 3 out here, and that's, so this is 9 and this is 18, so that's 17. So the 17th neuron that spiked in that order is also would be firing at the same phase or at the same time out here. You can do that with the next column. So number 22 also fired, and you know, all the others fired. So if you take a particular sliver of time and look at which are the neurons that fired and put those, you know, just assign a number to each of these neurons that fired from 1 to 81, you just put it in the respective blocks out here, and you will see that they're all 3s, right? And if you're looking for 6s, you can do the same thing out here, right? All the 6s would be sort of labeled in this manner, all the 3s and so on. So what you have in this particular solution is that a set of neurons fire together, then another set fires, then a third set fires, and so on till you go through all 9 groups of neurons, and then you start at the first. So you have an oscillation that goes through all the numbers, all the places in Sudoku, and repeats itself, right? So what you end up with out here is a solution that is constrained by the coloring of this network. All right. Now I can start at a different initial condition, or I can zap the network, perturb it, and it will go, in this particular case, it will go to a different Sudoku network. It turns out that there are a lot of nuances to the solutions out here. The symmetries of the solution matter as to which is more stable, which is less stable, and all of that. But we can talk about that later if you're interested. But for now, let's just say that you can have the network visit different colorings of the graph, each corresponding to a Sudoku solution, right? So here's the cool part of this, right? You start off with a network consisting of 81 neurons, all right? And the goal is to generate spatiotemporal patterns, because what did we say? We said a particular spatiotemporal pattern can be mapped to a particular behavior, right? So if you're thinking about olfaction, you want to have a large number of spatiotemporal patterns because you want to be able to smell a lot of different things, right? You want to be able to distinguish between lemon and orange, even though both are citrusy, and you want to have a large number of different possible smells that you can recognize. And there are some experiments, not necessarily accurate, but there are some experiments that have suggested that humans can smell 1 trillion odors. Turns out that that isn't true. At least in my reckoning, it isn't true. But how do you store a large number of patterns using a small network is one of the things that one can talk about. So if you look at a Hopfield network, and I think you guys looking at the syllabus, you've been through a Hopfield network, which is essentially an associative memory, right? So you have fixed points in your space, and depending on where you start, you're going to go and end up in one of these fixed points. Now, the capacity of networks like that is, at best about 10% or 14% of the total number of neurons that you have. So if you start off with 81 neurons, you'd be able to put in about 10 memories out there. But in this particular case, you can put in a lot more in terms of memories, right? Because the memories are not stored as fixed points, they're stored as something that evolves in time. They're stored as an oscillation, and these oscillations are stable because we try to perturb them, and with small perturbations they'll still come back to the same oscillation. So rather than store it as fixed points, if you store it as cycles, because you have extended your space and time, you can, because you have 1 trillion Sudoku solutions, in principle, you can store 1 trillion memories using a really small network. All right. Now,
So, Collins, I have a question.
Yeah.
So, I understand that the points which are firing at the same time have the same number. But how do you figure out it's a 3 and not the 4?
You don't have to, because you can assign an arbitrary number.
Okay.
It wouldn't
It's not
matter.
correct.
No.
For example, it may not agree with the particular clues that are already given.
So this is not based on clues. So there are no clues, right? So the constraint is that you have a clueless Sudoku network, and all of the states, all of the colorings are valid solutions, right?
Suppose you have clues.
See, we haven't. So putting in clues and getting to a solution is a lot harder. And so these are the nuances that I'm not talking about. But it turns out, and this is what Pratyush showed, that if you have an oscillatory drive and you can introduce clues as different DC inputs to the neurons, then you can drive it to specific states. So Sitabra raises a really interesting point out here, and the reason it's important to maybe address is that how do I a priori figure out which solution I'm going to land in? Right? So this is a clueless Sudoku. If I had clues, could I take it to the unique solution or not? And that turns out to be a reasonably difficult problem to sort. We've done certain shady things to get it to the solution that we want, but you have 1 trillion solutions. Your phase space is riddled with these cycles. How do you know that you can get to a particular cycle? So in this formalism, what we're doing is we just give it a random input, it lands where it may, and that's going to be a Sudoku solution, right? But we don't have a way to say that I want to go to this particular Sudoku solution or that Sudoku solution using this network. It turns out that if you introduce oscillations and if you give inputs to the neurons such that different neurons get different amplitude of inputs, then they fire at phases that are determined by the amplitude of the input. So if you give it a high amplitude, it'll fire at an earlier phase. So we can actually arrange neurons to fire at specific phases by giving it specific DC inputs, and when you do that, we can force it to go into a particular solution. But yeah, so that's an interesting point to work on. All right.
Have you thought of doing the TSP in this formulation? Like, for example,
the traveling salesman problem?
Yeah, so for example, thinking of each trajectory as a arrangement of neurons.
Right.
Each neuron in a particular city,
Right.
in a particular phase arrangement, and then feeding in the
How do we go from
constraints, feeding in the constraints in the form of this DC currents.
So we haven't done that. We haven't done the traveling salesman problem. And one of the reasons that we haven't done it is really our ability to control which solution it will go to know what are the inputs, right? Because it's the phase space is really pockmarked with different possibilities, right? And so it's very hard to get there. But now that we have some control over it, that's one of the things that we want to do is apply it to specific problems, perhaps through a learning rule, to see whether we can drive it to solutions by penalizing DC inputs based on how far you are from the solution and so on. But yeah, so all interesting questions and things we haven't gotten to as yet. All right. Okay, so there's your Sudoku solution. Okay, now that's the tangent that I wanted to go to, the idealization of the olfactory system. And now I'll try and get back to the olfactory system, and let's look at real olfactory systems. Now, one of the olfactory systems that I sort of work with, a historical accident, and also because it's a very simple model system, is that of the locust. This is the American desert locust, and there are Joby in University of Hyderabad has been doing some excellent work looking at grasshoppers. They're easier to access out here, and doing some stellar measurements from grasshoppers. So here the equivalent of the nose in your locust is the antenna. The antenna has olfactory receptor neurons, and let's see. So there are olfactory receptor neurons that are present on the antenna. So if that's the antenna, you have a bunch of olfactory receptor neurons. So what happens is that an odor molecule, a volatile molecule, goes and lands on a receptor on these olfactory receptor neurons. It then leads to a whole cascade of events that can eventually result in a spike, and that spike then goes to the next layer, which in insects is called the antennal lobe, and in mammals it's called the olfactory bulb. So it's further down from the antenna to the olfactory bulb, and in the olfactory bulb you have a dense recurrent network of neurons, right? And what are the kind of neurons you have? You have a bunch of inhibitory interneurons, right? And the inhibitory interneurons are connected to each other. They extend their arbors across the entire antennal lobe, each of the lobes on either side. And so it's a dense recurrent network. And they also extend processes to these neurons that are known as excitatory projection neurons, which in turn also drive the inhibitory neurons, right? So inhibitory neurons connect to excitatory neurons and to each other, and the excitatory neurons connect back to the inhibitory neurons. But what's interesting out here is that the excitatory neurons don't talk with each other, all right? So a lot of the recordings that you see in the literature, in the insect olfactory literature on the antennal lobe, is going to be recordings of these projection neurons. And the reason it's important is because these are the neurons that extend further out into the equivalent of the olfactory cortex in mammals, right? So it's the mushroom body. So these extend out to the mushroom body. So what people have done is, over many years they've been looking at the activity of these projection neurons and trying to figure out what would happen if I throw a particular odor onto the receptor neurons. How would the projection neurons fire, right? So what you have out here are you take a particular projection neuron, you give it throw a mint odor at it, and there are multiple trials out here. You will see for mint, it generates these 2 bursts of spikes. For apple, it generates a single burst of spikes at the start. For strawberry, it fires all over. This is a second neuron, so you sort of impale a different neuron out here, and you will see mint generates a burst. Apple, instead of firing at the start, the neuron will fire at the end. For strawberry, or in this case octanol, you have 3 bursts of firings and so on. So here's where one comes to the connection between a spatiotemporal pattern and the odor, right? So if you take a bunch of different neurons, each of these neurons will fire or not fire in a characteristic manner for a particular odor, right? There'll be spikes at a given point in time, maybe it doesn't fire, and so on. So if you look at the entire population of projection neurons, you give it a rose odor, that population of projection neurons, some are going to fire, not fire, fire at a different time, and so on. So you can think of the activity of these neurons as sort of like a movie that spreads over time, right? And the question is, what are the frames of this movie? So if you look at the entire, if you look at the average activity of all of the neurons, what you will get is an oscillation, right? So it's been argued that these oscillations, in each of these oscillatory cycles, different neurons, different groups of neurons fire. In the second cycle or the third cycle, another group will fire, and then another group, and this extended movie is the representation of a particular odor, right? So an odor is represented as a pattern that evolves over time. Now here's the conundrum, and you know, there are a lot of experiments that talk about it, there are a lot of controversies and so on, but for our purposes, we'll think of a spatiotemporal pattern as a representation of an odor. Now notice the, yeah.
So, can you go to the next slide? So, is this essentially equivalence of the glomerular activation pattern?
Right. So the glomerular, so the locust, the projection neurons are multiglomerular, right? So unlike in other, right? But yeah, you could think of it as glomerular activation patterns. So okay, so here's the input that this that you throw at the antenna. So if you look at mint, this line that you see out here is the time over which the antenna is exposed to the mint odor at a specific concentration. That line is the apple odor that's exposed, right? But
So, does it help in any way for an OSN to be multiglomerular, or it's the other way around?
you know, we have thought about this question, and one of the answers that we have come up with is that if it is multiglomerular, it's better as a generalizer. So it actually has a broad swath of odorants that it'll classify as, yeah, not so specific to the odor itself. Yeah. So you know, typically in experiments you have this DC input that's going to the receptor neurons. But if you look at real life, there's a spectacular illustration of a rose and a fan that I made, and that's the odor plume that it generates, right? These are so you know, the rose is like a complex mixture of molecules, and it's wafting through the air, riding on this chaotic plume that goes through it. And the locust, if it sticks its antennae into the plume and tries to figure out what the odor is, the input that's going to the receptor neurons is not this DC smooth input. It's actually this pulses of odors that are coming in, right? So it's a completely arbitrary temporal pattern of odors, right? Now the second observation, which is this really spectacular set of experiments from Gilles Laurent's lab, is what they did is they managed to give an odorant in a really controlled plume. So you have the odor just going along this line out here, right? So it's a very controlled plume, and you have a locust that's hanging out in the neighborhood. Now, one would think that if the locust wants to figure out, so this is a experiment where the locust has to figure out what the odorant is, what it would do is it would stick its antenna into the odor, hang out over there till it's figured out what the odor is, and then just quit, right? But instead, what the locust does is that it intermittently samples the plume, right? It doesn't give itself a DC input, but it goes in and out of the plume. And one wonders why that would be the case, right? You'd never get a locust just sticking its antenna in and getting it out. So if there is no intermittency, and I'm using the word intermittency rather loosely, if there is no intermittency, if there's no variability in the odor, the insect uses its behavior to inject intermittency or inject variability into the sampling of the odor. Much like when we smell stuff, if it's a new odor, you'd probably have a respiratory cycle that's a little faster and so on, right? So we tend to inject variability into the stimulus if such variability is not there. So perhaps this variability is a feature, not a bug of the network that looks at odorants. So that's the kind of problem that we try to solve. So what are the neural invariants of an odor, or why does a rose always smell the same? And you know, the idea being, of course, so far we've been saying that there's a spatiotemporal pattern, time matters, right? And the spatiotemporal pattern is what represents a rose. But now you can think of this movie as coming in spurts, all jumbled up together, and you can still say, oh, that's Cholet, that dates me. But you know, that's a terrible example, but but nevertheless, right? So you've given, you're given this spatiotemporal pattern of images and sounds, except in small spurts and shuffled, and you can make global sense of it, right? So how do you actually do that is the question. Yeah.
When you smell rose, it doesn't have to be specific, the same rose you smelled before.
Right.
It will be a range of, rose smells.
Sure.
It's an approximation at the end, right?
Right.
Maybe
But
categorization.
okay, but let's take let's say you're smelling the same rose. The point out here is that even if you're smelling the same rose, on day one it's coming on a different plume of air, on day two it's coming on a different plume of air, so there's no way to say that it is the same rose. All right. Yes.
So if I look at the antennal tip position, there are some positions where it's sticking to the odor, but it's not having any signal.
Oh, you know, this is activity of a projection neuron. This is not a receptor neuron. So this is one synapse downstream from the receptor neuron, and that projection neuron can fire at different times. So if you look at sort of the average activity in the antennal lobe, you can, or in the vicinity of that neuron, you can see that there are these little bumps, but there's a certain perhaps stochasticity to whether it's going to fire or not, right? So that's actually an interesting point that you bring. This is one projection neuron. So there are hundreds and thousands, but one would think that if that projection neuron fired once, right, it should fire a second time too. But there is a stochasticity to whether it fires or not. There's also, it does fire, but it doesn't fire at all the points in time. You can see that the neuron sort of trying to fire around here, but it doesn't actually get to fire. And there can be many reasons why that happens. But anyway, there is a certain stochasticity to it. Sitabra, I think I'm sort of going way over time. How much can I break the rules?
Still not way over time. It's still not at level. your official time is 11 and you can take a few more minutes. Right?
Oh,
Which
just
is a natural stopping point.
Okay, good. Given that things are lenient, we might go on and on. Okay, all right. So the question that I'm trying to look at is what are the neural invariants of an odor in this really turbulent plume? What is it that remains the same, such that I can always say that it's a rose? So in order to do that, we of course constructed an antennal lobe in a computer, and in the locust there are about 300 inhibitory interneurons and 900 projection neurons, and people have stuck an electrode so many times into the antennal lobe of a locust. If it's stereotypical, they've certainly oversampled the number of neurons that you have. So you have inhibitory neurons that are reciprocally that inhibit each other, and you have projection neurons that drive inputs to this. So the way we model an odor is that different projection neurons will get randomly picked projection neurons will get an input for a particular odor. For a different odor, it can be an overlapping but different population of projection neurons. Inhibitory interneurons, on the other hand, we know sort of extend very broadly, and they get input from almost the entire population of receptor neurons, and so they have very broad so almost all the inhibitory interneurons get about the same input regardless of what the odor is, because that's what we think happens in the antennal lobe. Now, if you look at the activity of neurons in the antennal lobe, here's a reciprocally connected group of inhibitory interneurons, and you will see that when one of these neurons fires a bunch of spikes, the other is silent, and when this spikes, the first neuron is silent. Now, the reason they switch is actually interesting. There is the concentration of calcium inside the neuron goes up with each spike, right? So every time a spike happens, there's more calcium that goes into the neuron. And locust neurons have channels that are known as calcium dependent potassium channels. So when there's more calcium, more of these potassium channels open, which sort of drives potassium out, and it makes the neuron more hyperpolarized. So what you'll see out here is that the first two spikes are close together, and as you go further down the spike, the time between spikes increases, right? And the reason for that is this calcium dependent potassium current. And at some point, the spikes are so far apart that one of the neurons is not able to suppress the other neuron sufficiently, so that revolts and takes over the country, and then you have that. Sorry, I was getting carried away. I just read the newspaper, but anyway. And the other neuron sort of fires, and if you turn off this calcium dependent potassium current, or you turn off the calcium, only one of the neurons will fire and the other won't, right? So it's a winner take all kind of a thing. Okay, now this is what we saw before. If you have groups of these neurons connected together, they'll fire in alternation, much like your motor systems that we talked about. Now, here's an interesting thing, right? So let's say you have these three groups of neurons, inhibitory interneurons, that are connected to each other, and I have projection neurons that excite these different inhibitory interneurons, and when I perturb one of these, so what would happen if I didn't have these excitatory neurons firing? Right? So I can run the system such that only one of these inhibitory neurons would fire and everything else would be silent, right? Winner take all rather than this oscillatory dynamics. Now, if a projection neuron if a projection neuron fires, it's going to excite this inhibitory interneuron. And what will happen is that the activity will suddenly switch to that neuron till the next perturbation comes, right? So you have essentially a bunch of switches, and the excitation is kicking these different switches one at a time, right? So that's the kind of thing that we do. So you have one group of LNs firing. When a projection neuron fires, then you kick it, then there's going to be these switches that are happening all across, and these are basically perturbations that come in from the projection neurons, right? So for this simple all-to-all connected network, that's a rather easy thing to achieve, right? But what happens when you have a far more complex network where this is the adjacency matrix? So there are 30 neurons. So this is a 30 by 30 adjacency matrix. The yellow dots are the ones of this adjacency matrix. It tells you which neuron is connected to which. And when you give it arbitrary perturbations, you see certain switches happening, but you don't quite know what's happening in general. There's no easy way to wrap your mind around this. So how do we go about figuring what's happening in this particular network, right? And this sort of brings us to I guess some of the things that you've been looking at in this workshop, right? How do you look at connectivity in networks? And one of the ways to look at connectivity in networks is modules, right? So I don't know, have you covered network modules? Right, great. So here's an example from Newman looking at the connectivity structure of these networks. So here are groups of neurons that are densely connected to each other, and across groups the connections are not quite as dense. You can partition a network, arbitrary networks, based on their connectivity, right? Now this would all be great if I were looking at excitation, right? So if all of these connections were excitatory and these were densely connected within, I'd get something like this, okay? But what I have is an inhibitory network, and I want to figure out which are the neurons that will form communities that fire together, right? So rather than look at nodes that are densely connected, I want to form communities of sparsely connected groups, right? That's the challenge that we have out here to figure out. So it's like saying that you only put your okay, that analogy didn't work out in my head, but I was thinking friends, enemy, it didn't work out. I need to think about that a bit more. But anyway, what you need is you need to make communities of people or communities of neurons that don't talk to each other, right? That's our inhibitory groups. So how do you go about doing that? So here's what we did. We basically just took your inhibitory network and all the nodes that weren't connected. So you flip the adjacency matrix such that the zeros become ones and the ones become zeros. So you have the complement, except there are no self connections, right? So the diagonals are always zero. So essentially you flip the matrix, and in the flipped matrix you find the communities, right? You find the communities once you look at the complementary matrix, and then you flip it back again, right? So essentially what you're doing this is a random network with about maybe 20% connectivity. Now what I've done out here is I take this adjacency matrix and flip it so that now the zeros are ones and the ones are zeros. And then I apply Newman's clustering algorithm or some other algorithm to this flipped matrix, and I'll generate communities I'll generate densely connected communities in this flipped network. And then of course I go back to my original network, and you will get two blocks that don't really talk to each other, right? So what we know out here is that we have, from this random network, we have two blocks that largely don't talk to each other, but across blocks you have a lot of connections happening, right? So they talk across but not within. So this is sort of not an approximation of coloring, but this is a way in which we can say that these groups inhibit each other across groups, right? So it's not it's not this very clear k-partite network, but it's some approximation of that. Now we can look at the activity of this particular network, and we'll organize it based on this particular rearrangement. And what you will see is that you will see these two blobs. That's basically the correlation between the activity of neurons in each of these groups that were classified as groups that didn't, neurons that didn't talk to each other. And you will see some higher amount of correlation. Now what you can do is you can look at the averaged activity between these communities of neurons that don't talk to each other, inhibitory groups. And when you do that, you'll see some sort of a pattern of activity emerging where, these perturbations tend to do about the same thing to each of these inhibitory groups. All right. So if you look at the activity in the space of the average firing rate of this group versus this group, what is each of these perturbation doing? It's basically making it jump from one state to the other, right? So now you have this large network, but we've broken it up into groups, and what do we get? We get one group active, another group active, and this sort of switches back and forth. Right? So you can think of it as a way of reducing the dimensions of the network. It's just that you're, rather than projecting onto principal components, you're projecting onto you're sort of averaging the activity of groups of neurons that fire in approximate synchrony based on a network measure that we arrive at. Okay. Now, the question, of course, is what is it that intermittent inputs are helping us do, right? So earlier we were just giving it some DC kind of an input. Now if we give it this intermittent input, which matches the statistic that you see when you look at locusts dipping their antenna into a plume of odor, you'll see that the odor comes on and off intermittently. It's just going on and off, and you have the activity of all the projection neurons. Now what we have done is we have done the same thing of figuring out what these inhibitory communities are, and if you look at these groups, you will see a very strong correlation that comes up between some sets of neurons, right? Now remember, this is a random network, right? This is not a network that I've built to have two communities or whatever, right? It's a completely random network. The communities are built using this particular community grouping algorithm, and you will see that you get these synchronized groups. So the activity and the degree of how well these correlations emerge really depends on the statistics of this intermittency, right? So if they go on and off, what we found is that the intermittency for locusts dipping their antenna approximately is in this range, and that's about where we get the most significant match to the kind of our ability to separate these clusters based on the dynamics of the network. So it does seem to serve a particular purpose, right? So going back to where we started, what are the neural invariants of an odor? So the hypothesis is this, right? Different odors stimulate different sub-networks of neurons, and the dynamics of the stimulated network basically reflects its topology, right? Because these groups reflect the sort of groups that are not connected to each other. That reflects the topology of this network, and this sub-network that's stimulated is what's invariant about the odor. So it's the dynamics in this case captures the topology of the network that was stimulated, and what we think is that it's the topology that's the invariant. So it doesn't matter how things, what's the order in which things come, if eventually over time, if you're able to figure out which is the subnetwork that was stimulated, you have your representation of a rose. The second thing is, so you can think of it like, different, going through the different states of the network, and if you give it sufficient time, it's going to visit all the different states of the network, right? So this is where intermittency plays a role. If you don't have these perturbations coming, your solution is going to go and sit in one of these places, right? And if it's still a DC input, it's just going to sit out there and it's not going to move. But once it sort of once it intermittently perturbs the network, it's going to kick the system to a different state, and then it's going to explore the entire state of possibilities for the subnetwork only if you're injecting intermittency, which is why we think that in order to explore the entire space that you get from stimulating a subnetwork. You need to do it with these perturbations that come in. All right. So the idea out here is the topology of the network is reflected in the dynamics, and by looking at the dynamics we can infer the topology, and it turns out that neurons one synapse further down are doing exactly that. They are inferring the topology of the network. So to put it all together, active sampling is a way to explore the states of the network and fill in the details of an order representation. Just give me a second. I just want to say that I finished on time or thereabouts. So the structure of the network is actually the invariant code. I'm not going to talk about the last bit, but I'll stop here. Yeah. What's the question you had?
So isn't that similar to solving a Sudoku board which has some information filled in it? So isn't what that so it won't visit all the networks, it'll only visit the ones which correspond to this what is present in the stimulus.
Yes, exactly. So in the case of Sudoku, there were this huge set of possibilities. Out here, of this huge set of possibilities, think of it as you're just looking at a sub-network of your Sudoku network, and it's going to visit the colorings of that sub-network. That's what.